During this Genetics 112 lab, we mainly focused our experiments and results on sourdough starters. Before getting to exact details of how we participate in this experiment, I want to shine light on the brief question, what are sourdough starters? They are essentially relevant when it comes to sourdough bacon, are responsible for dough rise, and consist of two simple ingredients, which are water and flour. After the ingredients are mixed, it will be colonized by wild yeast and lactic acid bacteria, then fermentation will occur. Wild yeast forms bubbles, releases gas, produces alcohol, and is responsible for increasing the volume in the starter and bread rising. Lactic acid bacteria produces lactic acid and is responsible for aroma and texture. Now on to the lab procedures and experiments. Every lab session, when we further conduct experiments, we're maintaining this question in the back of our heads. Are we able to produce a starter that is resistant to osmotic stress and still perform its intended function? Now, each pair of students had to grow their own sourdough starters in test tubes. Generally, one starter was the control sample, which consists of water and flour, while the other starters was the salt sample, which consists of water with a given salt concentration and flour. This given salt concentration varied in each pair of students. We selected the sodium chloride concentration with a molarity of 0.125. We had the lowest concentration out of all the pairs, with the highest concentration being 1.5. We had four different experiments to one, grow our sourdough starters in different conditions, and two, grasp more features with plate streaking, PCR amplification, cell electrophoresis, sequencing following Sanger sequencing, and automated DNA sequencers. During our first two experiments, we grew microbes from the starters, one given by the instructor and the other selected microbial colony from our first plate onto two separate plates. This allowed us to isolate colonies of microbes, which are later used for PCR amplification of genes of interest. But before moving on to our four rising rate experiment graphs, which are graphs that we constructed based on each sourdough starter experiment, I want to quickly demonstrate our procedure of plate streaking. When streaking our first plate, each partner was given a plate labeled YPD or TSA with a shared amount of starter given by the instructor. With a sterile toothpick, students were to streak the starter in this motion. Each partner were to continue this motion at a different direction with a new sterile pick for a total of 3 times. In the end, it should look like this. Now onto our rising rate graphs. This is our rising rate experiment 1 with our sourdough starters, one being the control and one being our salt concentration. We were asked whether a harsh environment, in this case a salty one, will hinder the sourdough starter to grow or have no effect at all or provide benefits to the microbial community. The basic idea is harsh environments ultimately make it difficult for the organism to live. As expected, the sodium chloride concentrated environment slightly hindered the growth of the sourdough. As shown in the graph, the salt sample did not grow at all during the first 4 hours. It was after when it suddenly started growing parallel to the control sample. It can be concluded that the microbes had trouble adapting to the environment and creating their initial colony until it surpassed 4 hours. Now, based on this graph of all the pairs discovered of the first experiment, it's unexpected that a highly concentration of salt, be it the 0.25 or 0.5 or the 1 mole molarity, provided less difficulties for the microbes to grow. The microbes grew noticeably more than in those higher concentrations, given the idea that more harsh environments equals more difficulties to grow. After the experiment, we were given the idea of what if we had a sourdough starter that has lived in salty conditions for 2 weeks and we fed it salt water. Will that starter already have gained resistance to sodium chloride and thrive better in salty conditions? Rising rate experiment 2. In this experiment, the general procedure has remained the same, but the experimental question has changed. Each group is given two different starters again. The control sample, which is a tube of sourdough starter that has been fed flour and water every single day. And the salt sample, which is a tube of sourdough starter that has been fed flour and sodium chloride water every day for the past 2 weeks. In this experiment, each pairs starters are not fed plain water. Instead, they are fed the selected concentration of salt water, in our case 0.125 moles of sodium chloride. These two starters are then compared to our instructor's two control tubes, one being a control sample, which has been fed only flour and water every single day, and our salt sample, or their salt sample, a tube of sourdough starter that has been fed flour and sodium chloride water for the past 2 weeks. But these sourdough starters are fed plain water. The question is, after the sourdough starter has grown in the presence of salts for over 2 weeks, has it evolved to gain resistance to salt and thrive better in these conditions? Our idea is that the salt sample that was fed sodium chloride for 2 weeks would have evolved enough to survive slightly better in salt conditions. Based on our graph, our salt sample, which is labeled in green, did not gain any noticeable resistance to salt. Our sample grew at the same pace as our instructor salt sample, which is labeled in red, that has been fed only plain water. They remained at the same pace until the 16-hour mark, where our salt sample started to maintain its growth at a constant rate. It can be concluded that the microbial community in both our salt sample and our instructor salt sample was extremely damaged due to living in salty conditions for 2 weeks. As shown in our graph, our control sample, which is labeled in yellow, which grew in favorable conditions for 2 weeks, thrived better, although it was fed salt water. The rising rate graph number 3 follows the same procedure as the previous experiment. Each pair is given again two different starters, the control sample and the salt sample, but in this case the salt sample has been fed flour and sodium chloride water every day for almost a year. Again, these sourdough starters were fed the selected concentration of salt water, in our case 0.125 mole. These two starters are again compared to our instructor's two tubes. We have their control sample again, and then their salt sample, which also been fed sodium chloride water every day for almost a year. These sourdough starters are fed plain water. Now the question is, how did things change between this experiment and the previous, and does long-term treatment with salt provide evolutionary pressure on our microbial community? The idea is, with more than enough time for microbes, they could essentially gain noticeable resistance to salt after growing in salty conditions for almost a year. Based on our graph, our salt sample, which is labeled in green, gained resistance to salt. It grew more than our control sample, which is labeled in yellow, and grew just a tad bit more than our instructor's control sample, which is labeled in blue, which was fed only plain water. Essentially, after a year, it gained enough resistance to grow normally, although it was fed salt water. It's important to note our instructor's salt sample, which is labeled in red, and its abnormally large growth. This could imply that after living in harsh conditions for so long, it held back so much production until a fresh wave of energy was introduced, in this case a favorable environment such as plain water. Rising rate experiment number 4 follows the same procedure as the previous experiment. Each pair is given two different starters, one being the control and the other being the sample. In this case, the sample is fed flour and sodium chloride water every day since 2022, September 2022 to be exact, a little more over than a year. Again, these sourdough starters are fed the selected concentration of salt water. In our case, is 0.125. These two starters are again compared to our instructor's control tubes, one being control sample and the other being the salt sample with the same procedure. These will be the sourdough starters that are fed plain water. Now the question is, as the previous question, does an even longer term treatment with salt provide more evolutionary pressure on our microbial community? And the idea is, of course, with even more enough time for microbes, they should gain an even more resistance to salt after growing in salty conditions for over an entire year. Based on our graph, our salt sample, which is labeled in green, grew at the same pace as our instructor salt sample, labeled in red. This pace continued until the 12-hour mark, where our salt sample started to gradually decline in growth. Because of this similar pace amongst our salt sample and our instructor salt sample, it can be implied that our salt sample gained little to no resistance to salt. Contrarily, the microbial community within the salt starters may have been too damaged to grow. This is further proven when both are compared to our control sample, labeled in yellow, and our instructor's control sample, labeled in blue, which have not been fed salt water since September of 2022 and grew significantly more. So, stepping away from the graphs, I'll now show you an overview of DNA sequencing and how we used it in our experiments. Specifically, what we amplified and sequenced, spoiler, ITS and 16S, and why is ITS and 16S sequencing used. Um, so, what is DNA sequencing? DNA sequencing is a determination of the accurate order of the nucleotides in a given DNA fragment. This is incredibly important in identifying the structure and function of the genes. Protocol involves different processes, including the isolation of desired DNA of an organism, the amplification of desired region of DNA using PCR, PCR being the making of many copies of desired DNA fragments, the separation of amplified PCR product done by gel electrophoresis, purification of DNA, serving amplified fragments as template for sequencing, sequencing done following Sanger sequencing, determination of correct nucleotide order done by automated DNA sequencers. Now, on our colony PCR1, we selected out colonies of interest from week 2 starter streaks to make many copies of the fungal ITS, which is internal transcript spacer region, from each selected colony from YPD plates, and bacterial 16S region from each selected colony from TSA plates. The ITS region we amplified is highly variable across different fungal species. The same can be implied. The bacterial 16S region is highly variable across different bacterial species. After sequencing these regions, we can determine the species level identity of the microbes growing on our plates. Right. These are the samples that were successfully sequenced. We have our 16SF plate 1, which is Saccharomyces cerevisiae, which is bacterial. We have our 16SR plate 1, which is lacticase Bacillus paracasea, which is also bacteria. And then we have our ITSF plate 1 and our ITSR plate 1, which are both uncultured fungus. These are the samples that were unsuccessfully sequenced. We believe the first unsequenced organism is Saccharomyces cerevisiae because the microbial colony looks that of that organism, very small, round, and mildly isolated. We believe the second unsequenced organism is Staphylococcus capitis because the microbial colony looks that of that organism, large dots and very condensed. After 2 weeks, the composition of the microbial community amongst each pair of plates has significantly decreased. The only organism that was successfully sequenced was Saccharomyces cerevisiae. That concludes our experiments. But now, what would I do to continue this project? My approach is simple. It's to look under a microscope. Although we can observe the physical appearance of the microbial colonies with the naked eye, it cannot be certain what organism they can be or be more related to, especially given the fact that these colonies were unable to be sequenced. An approach to understand 1. What these unsequenced organisms can be, and 2. How much damage a salty environment can have on the community will be conducting experiments under a microscope. The general objective for number 1 will be comparing the physical appearance of the unsequenced organisms to the organisms found in the original sourdough starters, including Saccharomyces cerevisiae, lacticase Bacillus paracasea, Bacillus cerecia, and more. Then for number 2, we will be identifying the problems salty conditions bring to the microbial community. Could an excess amount of sodium chloride prevent certain proteins through the cell membrane, or cause the cell membrane entirely to rupture, or introduce loads of mutation causing the cell to go to apoptosis? Well, that concludes my video. Thank you for watching and have a wonderful day.